Good afternoon, viewers. I am Dr. Y. Kumar, Assistant Professor on Contract Basis in the Department of Botany, Dr. BRA OU. I welcome you all to this teleconference session on advanced concept, plant cell structure and organelles and its function, which is offered by Department of Botany. Actually, this session designed to the UG students and PG students as well as aspirants who are prepared for the all different type of competitive exams. And we are privileged to have the esteemed expert having with us today deliver this session in detail. Before introducing them, let me give learning outcomes of this topic. After completing this session, you will be able to differentiate prokaryotic cell and eukaryotic cell and explain in detail about plant cell structure, plant cell wall, plasma membrane and its functions, and compare different type of cell organelle structures and functions, and evaluate metabolic activities of different cell organelles occurring within the cell organelles. And I am delighted to introduce today expert Dr. T. Srinivas sir, Head of Department of Botany and Control of Examination and SRR Degree College Autonomous, Karimnagar, and another expert Dr. T. Padala Tirupati sir, Department of Botany, SRR Degree College, Karimnagar. And viewers, the small announcement: if you have any doubts on these topics, you can contact over the phone. The contact number is 2368-0456. Once again repeat, 2368-0456. Now I request first subject expert Dr. T. Srinivas sir. Sir, actually our students bilingual, both English medium student and Telugu medium students. You can use the both languages. Sir, on the basis, the first question is, first of all, the cell history and overview of the cell and differentiate between prokaryotic and eukaryotic cell. Please explain about to our students.
Okay, sir. Good afternoon, viewers. I am Dr. Thirukavala Srinivas, Associate Professor of Botany, SRR Government Arts and Science College, Autonomous, Karimnagar. And coming to the cell, yes, cell is the fundamental structural and functional unit of every living organism, and the cell was first discovered by Robert Hooke in 1965 in the cork of oak tree, and he observed as a dead cells. The cell means it is Latin word and meaning of small little rooms, and he explained everything in his book, that is Micrographia. And second scientist Antony van Leeuwenhoek first saw and described a live cell in 1674, and he named as animalcules, and is a father of microbiology. And the third scientist Robert Brown, so he first observed the nucleus of cell in 1831 in orchid leaf. The major theory is the cell theory, and it is proposed by the Schleiden and Schwann in 1838 and 39, and according to them, the first principle is the all living organisms are composed of cells, and they are the structural and functional unit of living organisms. But this theory is not at all explain how the new cells were formed. Later in 1855, Rudolf Virchow explained his third principle, that is all cells are arise from pre-existing cells. That means కొత్త కణాలు అనేవి పాత కణాల నుండి సెల్ డివిజన్ ద్వారా ఏర్పడతాయని చెప్పారు. సో ఇది ఫండమెంటల్ థియరీ. దీస్ త్రీ ఆర్ ది మెయిన్ ప్రిన్సిపల్స్ ఆఫ్ సెల్ థియరీ. On overview of the cell, so here the cells are differ greatly size, shape and activities. For example, so mycoplasma is smaller cell, nearly 0.3 millimicrometer, and egg and ostrich is the largest isolated single cell, and nerve cells are some of the longest cells, and here the RBC or 17 micrometer is diameter, whereas bacteria is 3 to 5 micrometer, where virus is 0.02 micrometer. The cytoplasma is the main arena of cellular activities in both plants and animal cells in the living state. And over the ribosomes are non-membrane bound organelles found in all cells. The animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. So in this advanced concept in plant cell structure and organelle functions, so the specialized for some of the cells are specialized for photosynthesis, storage, support and other vital process, and these advanced studies in plant cell biology explore not only the structure but also molecular mechanism and signaling pathway and dynamic interactions between organelles. So the structure of prokaryotic cell. So the best example is bacteria and our cyanobacteria cell, and this is the containing cell envelope. So it consists of with three members, one glycolics and cell wall, plasma membrane, and bacteria can be classified into two groups based on the composition of cell envelope. And here Christian Gram developed a Gram staining process. From that we get the two types of bacteria, that is Gram positive and Gram negative, where Gram positive is in blue in color, where Gram negative is in colorless. And the second, the glycocalyx includes capsules or thick sheath or and slime layer, or that means it is in loose sheath. Coming to the cell wall, made up of peptidoglycan or muramic acid and present in the cell wall, and the cell membrane extensions are the mainly is the mesosomes. So they are the help in the cell wall formation and DNA replication, respiration, secretion, and increase the surface area. That means the mesosome is folded inside, and the chromosome contain pigments, that is blue green algae best example, and cytoplasm it contains ribosomes 70S and main DNA nucleide or genophore, plasmids, inclusions bodies present. Then flagella, it contains three parts, one is filament and the hook and basal body. So these three containing the carries the 9+3 organization. So in this prokaryotic cell represented by bacteria or blue green algae, mycoplasma, here the PPLO, pluriopneumatic like organisms, these are called as jokers of plant kingdom, and they may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, that is rod like, and coccus, that is spherical, and comma shaped are called as vibrio, whereas spiral are spirillum. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genome DNA, the single chromosome circular DNA is present, and the similar DNA are called plasmids or extra nuclear. So coming to the ribosomes, so these present in prokaryotic as well as eukaryotic cell, and this is first reported by George Palade, and these are made of two subunits, that is 50S and 30S, which when present together form the 70S, where in eukaryotic those are the 80S, that means 60S, subunit and 40 S subunit. Here S means Swedberg unit, that is sedimentation coefficient, is directly a measure of definitely and size. Rhizomes are the site of the actual protein synthesis. Here several ribosomes are attached to a single mRNA, form a chain polyribosomes and or polysome. These ribosomes of polysomes are translate the mRNA into the protein synthesis. And here some of inclusion bodies are also present in this bacteria. These are the reserved food material in prokaryotic cells. Those are the phosphate granules and cyanophysin granules and glycogen granules. And some of gas vacuoles are also found. And eukaryotic cells have, where eukaryotic cells have membrane bound nucleus, double membrane cell organelles, and prokaryotic cells they do not contain.
Sir, okay sir, you explained well about the prokaryotic cell and history of the cell and overview of the cell. Sir, what are the some differences between eukaryotic cell with from the prokaryotic cell?
Yes. So here the prokaryotic cell and eukaryotic cell. So here prokaryotic cell means the best example is bacteria and cyanobacteria, and where eukaryotic is any plant cell or animal cell.
Yes.
So first the prokaryotic cells are they are singular in simple and single unicellular, whereas eukaryotic is a complex and the multicellular. In prokaryotic, the genetic material is in the form of a nucleide. Here the nucleus, true nucleus is absent, that is nucleus membrane is absent, wherein eukaryotic the true nuclei, that is a membranous nucleic material, that is DNA, is present. And here there is no membrane, two membrane bounded organelles, and whereas in eukaryotic many membranes are present, membranes are present, and the prokaryotic cells are called as a very primitive, and eukaryotic is very advanced. So this is the main difference between prokaryotic and eukaryotic cells.
Okay sir, thank you. So prokaryotic very simple cells. So eukaryotic some more complex. So in terms of cell organelles, because in prokaryote, if you take prokaryote, only one cell organelles are there, that is 70S ribose, as you explained.
Yes, sir.
And if you take the prokaryotic cell, sorry, eukaryotic cell, that is more complex because of having more cell organelles. As you said, membrane cell organelles, whatever it may be, nucleus, mitochondria, chloroplast, Golgi complexes, these are all. Right? Thank you, sir. Thank you very much. Now I request second experts, Dr. PADALA THIRUPAT sir. Sir explained about prokaryotic cell and eukaryotic also. Sir, just explain about in eukaryotic, differentiate between eukaryotic, that means plant cell, and animal cell in eukaryotic.
Okay, thank you Kumar sir. This is Dr. PADALA THIRUPATI, Assistant Professor, Department of Botany, SRR Government Arts and Science College, Autonomous Karimnagar. Today we are going to discuss about the plant cell and animal cell, differentiate between plant cell and animal cell. We know that the plant cell is made up of, it is surrounded by cell wall. Cell wall is the distinguished feature of the plant cell, but whereas the animal cell does not possess cell wall. This is the first and foremost important characteristic feature we can identify whether the cell belongs to the animal or even the plant. And the second most important identification feature of the plant cell possesses the vacuole. Central vacuoles are present in plant cell, whereas animal cell does not possess any vacuole. This is the second most important differentiate between the plant cell and animal cell. The third most important difference between the plant cell and animal cell, that is the plastids which are present in plant cell, whereas the animal cell does not possess. The final most important characteristic feature identification, the difference between the plant cell and animal cell, that is the centrosome. Centrosome present only animal cell, but whereas in the plant cell is absent. సెంట్రోజోమ్ అనేది కేవలం నిజకేంద్రక కణమైన జంతు కణంలో మాత్రమే ఉంటుంది, వృక్ష కణంలో ఉండదు. ఇది అత్యంత ముఖ్యమైనటువంటి మనకు గుర్తింపుకోవాల్సినటువంటి ముఖ్యమైనటువంటి భేదాలు.
థ్యాంక్ యూ సార్, వెల్ ఎక్స్ప్లెయిన్డ్. సో plant cell and animal cell.
Yes.
If you take the any cell, plant and animal cell, some are the major or similar components are there.
Yes.
The common components, if you take the any cell, for example, plant cell mainly the cell wall, next followed by plasma membrane, next nucleus,
Yes.
and double membrane cell organelles, and other single membrane cell organelles are there.
Yes.
Only major difference between the animal and plant cell, except the cell wall absent in animal,
Yes.
cell wall present in plant.
Yes.
Right. Just in brief detailed about the cell components. First cell wall, after that plasma membrane, after that nucleus, mitochondria, and chloroplast.
Yes.
Next followed by the
cell
single membrane cell. That their structures and functions.
Yes. So, then afterwards we will going to discuss about the cell wall. It is the most important characteristic feature of the all plant cell. This is a first and foremost whenever the plant cell is going to divide during cell division or mitotic cell division, the central middle lamella which is produced during cell division, this middle lamella is made up of calcium magnesium pectate. This is the most important chemical composition which is definitely present to make this middle lamella. So, then afterwards the primary cell wall is synthesized. This primary cell wall is very simple and the flexible nature, which is made up of cellulose, hemicellulose, even though the pectic substance also present in primary cell wall. Then whereas then afterwards, after developmental stages, the secondary wall is also synthesized. This secondary wall is consist along with cellulose, hemicellulose, pectic substance, the other lignified or lignification process has completed. Due to that reason, the secondary cell wall is a dead nature due to the high amount of the lignin is composed, or even though ఈ లిగ్నిన్ అనేటువంటిది సంసేవితం కావడం వల్ల ఈ ద్వితీయ కనకవచం అనేటువంటిది మనకు దృఢంగా ఉంటుంది, గట్టిగా ఉండడంలో తోడ్పడుతుంది. డ్యూ టు దిస్ సెల్ వాల్ మేజర్లీ all plant cell they can sustain, they can survive due to extreme conditions like hot summer, then cold regions like northeast region, the hilly regions or mountains or the Himalayan ranges, the plant can going to sustain or survive. Even though the tropical region, especially in Africa continent or the equatorial zone, the plants can sustain more than 48 or 50 degrees Celsius due to the presence of the cell wall. This is the most important structure. So, then what is the advanced concept of the plasma membrane? The cell wall. The cell wall, it is not only made up of the cellulose, hemicellulose, pectin, but also it is involved in the cytoplasmic adjacent cells and enabled intercellular communication and transport. And also it is most important to role of the stress responses. What are the stresses? We know that the water stress, salt stress, heat stress, temperature stress and cold stress, this type of conditions the plant can sustain and survive due to the presence of the cell wall. The cell wall proteins and enzymes remodel structure during even though the pathogen attack. Whenever the microbes like bacteria and the viral and other fungal pathogens, whenever they are going to attack the plant, the cell wall can sustain, the cell wall can protect from the microorganisms. So, then afterwards the cell wall, the cell membrane is surrounded on the cytoplasm. The cell membrane is the best nature, that is the fluidity nature. It is made up of the phospholipids or lipoprotein layer. Cell wall is made up of cellulose, hemicellulose, pectin and lignin, but whereas the plasma membrane or cell membrane is made up of the lipoprotein layer. Lipids and proteins which are present in the membrane. So, that is why the cell membrane or plasma membrane is ability to fluidity nature, or the cell membrane also most important to the internal to external to internal pathway to transport or translocation of the some of the molecules related to proteins and other molecules also, they are moved towards the internal part of the cell. And the plasma membrane also controlling movement of the substances. The receptor mediated signaling also we are observed, that is nothing but hormones impact, even the defense mechanism, and signaling pathways also we are observed in the presence of the plasma membrane. And the membrane micro domain also present these lipid wraps. For example, for organizing proteins, it is involved in vesicle trafficking between the endomembrane system. These the slide we are showing, that the colorful picture we are observed, that is the most important model, that is Singer and Nicholson has given the Cauchy fluid or the fluid mosaic model. ఫ్లూయిడ్ మొజాయిక్ నమూనాను మనకి ఇచ్చినటువంటి శాస్త్రవేత్తలు సింగర్ అండ్ నికోల్సన్ అనేటువంటి ఇద్దరు శాస్త్రవేత్తలు ఈ ప్లాస్మా త్వచానికి సంబంధించినటువంటి పరిపూర్ణమైనటువంటి ఆమోదయోగ్యమైనటువంటి మనకు నమూనాను ఇవ్వడం జరిగింది. అంతకుముందు ఇచ్చినటువంటి శాస్త్రవేత్తలు మనం గమనించినట్లయితే అది ద్విత్వచ నమూనాని, అంటే డైలామెల్లర్ అని, ట్రైలామెల్లర్ అని, యూనిట్ మెంబ్రేన్ మోడల్ అని, అంటే మనకు ప్రమాణ త్వచ నమూనా అని, అదేవిధంగా శాండ్విచ్ మోడల్ అని, వివిధ రకాల నమూనాలు ఉన్నప్పటికీ కూడా మనకు ఆమోదయోగ్యమైనటువంటి ప్రస్తుతము మనం అనుసరిస్తున్నటువంటి నమూనా ఏదైనా ఉందంటే ద ఫస్ట్ అండ్ ఫార్మోస్ట్ మోస్ట్ యాక్సెప్టబుల్ మోడల్, దట్ ఈస్ fluid mosaic model has given by Singer and Nicholson. So due to that reason, the plasma membrane can ability to accept due to the diffusion, or even though the facilitated diffusion, or even though the active transport. The diffusion, we know that the presence of the concentration gradient from higher concentration to the lower concentration without involvement of the any energy or ATP, the molecules or ions, they can across the membrane from outer side to the inner side of the cell. Then what about the facilitated diffusion? The across the membrane by the involvement of the carrier proteins. The carrier proteins also we can call as porins. The porins are the special type of proteins which are accepting or passages, acted as a passages to the inner side of the cell. It is also we can call as facilitated diffusion. In this facilitated diffusion, we can also observe only one molecule can enter into the cell, or even the cell to the outer side, that we can call as uniport model. Then second type of model, that is we can call as antiport. Anti means both the molecules which they can across the membrane in opposite direction manner, that we can call as antiport. Then the final port, that is we can call as symport. In the symport, two molecules which are enter into the cell, or even the from inner cell to the outer cell, the same direction, that we can call as symport. So these antiport, uniport, symport, due to the presence of the carrier proteins, that we can call as facilitated diffusion. Sulabhatara visarana అని చెప్పుకుంటాం. There is no need any type of energy, ATP energy, for this facilitated diffusion. And my dear friends, remember, the facilitated diffusion no need any type of energy, but concentration gradient is more important. From the higher concentration to lower concentration, that is mandatory. Then third one, that is the most important, that is active transport. In this active transport, the energy is needed, that is we can call as adenosine triphosphate, ATP is necessary. This ATP is acted as a pumps. These pumps are involved the opposite direction to the concentration gradient, which enter into the from the lower concentration to higher concentration. This is nothing but the osmoregulatory process. This osmoregulatory process we are observed the across the membrane in inner side to the cell. Then we are going to discuss about the major cell organelles or the endomembrane system, that is the first and foremost important cell organelle, that is nucleus. Sir, I already told that the nucleus also we can call that is a cell brain. Why we are called nucleus is a cell brain? Because the nucleus is controlled all type of metabolic and physiological activities of the cell organelles, including the control of the molecular level and the each and every organelle under the control of this nucleus. So that's why nucleus we can call as central cell. Nucleus also we can call as the cell brain. And the most important cell organelle, it is most important to transfer of the heredity hereditary material from one organism to another organism. This nucleus has discovered by Robert Brown from the orchid leaf in 1831. It is a double membraned structure. We can know that is we can call as nuclear membrane, whereas the prokaryote does not possess this nuclear membrane. That's why the prokaryotes does not possess any definite nucleus. But whereas in all eukaryotic organisms, they are clearly absorb this nuclear membrane. It is a double membranous cell, double membrane, and the presence number of pores which are involved the translocation of the even though the mRNA from nucleus to the cytoplasm, and also some of the peripheral proteins also arranged on the surface of the nuclear membrane. Even though the inside of the nucleus we are observed one or two nucleolus also we are observed. These are observed by the especially Fontana specialized structures we are observed inside the nucleus. Inside the nucleus, the thread-like structure we are observed. The thread-like structure we can call as the chromatin. Chroma means the color body. Thread means a small hair-like structures that we can call as chromatin thread, which is also most important by the presence of the DNA and the RNA and other histidine and also the histone proteins that we can call as the basal proteins, which are involved in the genetic material. It is involved in the it is transferred from the parental to their children, their offshoots. Then the most important structure we are observed during the cell division, that is chromosome. This chromosome, whenever the chromatin thread is going to condensate to form the chromosome. Chroma means the color, zoma means body. Because the colored body we are observed during the cell division, whether the mitosis or even though the meiosis, we are observed the center part of the nucleus. Whenever the karyokinesis, the chromosomes are observed. The chromosomes are present, which is in different shapes during the cell division. That is anaphase, we are observed the 4 different types of the chromosomes, that is metacentric chromosome, submetacentric chromosome, acrocentric chromosome and telocentric chromosome also. Whenever we are observed the metacentric chromosome during anaphase, it looks like a V shape. Then whereas the submetacentric we are observed in the during anaphase, it looks like a L shape. And the third one, acrocentric chromosome, whenever we are observed during anaphase, it looks like a J shape. Then last one, telocentric, that it is I shape, or there is no any other part of the chromosome. But whenever we are observed the centromere is most important. Centromere is most important part where whenever during cell division, the chromatids are going to break down from the part of the centromere. The centromere is surrounded by the kinetochores. These kinetochores are most important to attach the spindle fibers. The spindle fibers are going to condensate, or even though the contractile during the anaphase, the chromatids are moved toward both the poles. And after centromere, the another contractile we are observed that we can call as a secondary contractile, or in that area the knob-like structure we are observed that we can call as satellite. The satellite present chromosome we can call as SAT chromosome. This SAT chromosome or satellite chromosome, which is useful for the giving the nucleus region. This is also we can call as NOR region, NOR region. NOR region means nucleolar organizer region. Then afterwards, the most important structure Audet has discovered, the most important structure, that is nucleosome. This nucleosome is a beaded-like structure when we are observed. పూసల వంటి నిర్మాణాలు మనకు ఏదైతే దారపు పోగుల మధ్యలో పూసలు ఏ విధంగా అయితే అమర్చబడి ఉన్నాయో ఆ విధంగా ఈ క్షార ప్రోటీన్లు అనేటువంటివి మనకు ముఖ్యంగా హిస్టోన్ ప్రోటీన్స్ అనేటువంటివి 8 ప్రోటీన్లు హిస్టోన్ ఆక్టోమర్ అనేటువంటి నిర్మాణంగా మార్పు చెందుతుంది. అకార్డింగ్ టు అవుడెట్, ది హిస్టోన్ ఆక్టోమర్ ఆర్ ది formed. This histone octamer consists H2A, H2B, H3, H4. These are the 8 proteins. These are basal proteins or alkaline proteins which are important for the hereditary process. Then another two histone octamers or nucleosomes are connected by the linker DNA. The linker DNA consists H1A protein, H1 protein. H1 protein present on the surface, on the presence on the linker DNA which is connect both the nucleosome. So this nucleosome is most important in the hereditary process. Then the
Right.
another.
Okay, sir. Sir explained well about the cell wall and plasma membrane. So cell wall chemical different and plasma membrane chemical different, and ultrastructure of nucleus also completed.
Yes.
Just brief discussion about the cell wall components. Okay, they are majorly made up of cellulose, hemicellulose, pectins. Clear.
Yes. Yes.
And plasma membrane, which is made up of phospholipids and proteins. And sterols, carbohydrates also present.
Yes.
But so small the variation between the primitive cell and advanced, that means eukaryotic cell.
Yes.
As you said, if you as you said, in prokaryotic cell the plasma membrane, eukaryotic plasma membrane same phospholipid protein.
Yes.
But the small variation, sterols absent in prokaryote.
Prokaryotes.
Sterols present in
Eukaryotes. Yes.
And cholesterol also present, and also carbohydrate also present in plasma membrane of the eukaryotic cell. And what are the major differences between prokaryote nucleus and eukaryotic nucleus?
Yes, prokaryotic nucleus does not possess any definite nuclear membrane, even though the naked DNA we can call as nucleoid. The DNA we can call as nucleoid. Why? Because there is no definite nuclear membrane is present. So that is looks like a naked, open DNA that we can call as nucleoid. Then what about the eukaryotic DNA or eukaryotic nucleus that is completely surrounded by nuclear membrane. So that is the most important. The second most important, prokaryote cell wall does not possess, especially bacteria cell wall does not possess cellulose. But whereas the another most important chemical composition of the cell wall, that is N-acetyl amino muramic acid, or even though that we can call as peptidoglycan, that is the most important chemical composition we are observing in the cell wall of the bacteria. But whereas the another prokaryotic of the blue-green algae members, that is cyanobacteria, or నీలి ఆకుపచ్చ శైవలాలుగా పిలువబడేటువంటి సైనోఫైసికి చెందినటువంటి శైవలాలు కూడా మనకు ప్రొకారియోట్స్గా మనం పిలువబడతాయి. కేంద్రక పూర్వ కణాలుగా, కేంద్రక పూర్వ జీవులుగా మనం చెప్పుకుంటాం. ఈవెన్ దో the prokaryotes of the blue green algae members, they are possessed in the cell wall this specific structure, that is a pectic substance. This pectic substance, whenever going to dissolve in water, the trichome, it is in nature, mucilaginous nature, it is going to easily slime, it is మనకు జారుడు స్వభావాన్ని కలిగి ఉండి అది జిగట స్వభావాన్ని కలిగి ఉంటుంది. వై? బికాస్ ఎక్స్ట్రీమ్ కండిషన్స్ లైక్ హాట్ స్ప్రింగ్స్ అండ్ మోర్ దెన్ 80 degrees Celsius they can survive, and even the deeper oceans, the most extreme conditions like alkaline nature, even though acidic nature, they can sustain. Why? Because the pectin substance is present in the blue green algae members.
Right, so thank you. So the nucleus ultrastructure, the nuclear membrane. Okay, inside the chromosome present, and the chromosome ultrastructure, how the chromosome made up of also clear.
Yes.
So clearly the chromosome in eukaryotic, chromosome made up of histone proteins.
Yes.
Right, that model called as nucleosome. So in nucleosome DNA is wrapped by histone proteins. Right, H1, H2, H3A histone proteins.
Yes.
Sir, with these histone proteins may be absent, I think absent in prokaryotic.
Yes.
Right.
Yes.
So only these are present in eukaryotic.
Yes.
Right. So ultrastructure clear, sir. Actually the frequently in competitive exam and theory exam, the frequently asked about mitochondria and chloroplast.
Yeah.
Just give the in detail explanation about mitochondria structure and function, and chloroplast structure and function.
Yeah.
Thank you,
sir. Right, sir. You are Kumar, sir. Because the mitochondria and chloroplast are the most important cell organelles in all eukaryotes, even though the mitochondria present both plant cell and animal cell, whereas the plastids are chloroplast which are present only the plant cell. Both the cells are most important. First of all, we will going to discuss about the mitochondria. This mitochondria first discovered by Kolliker, then afterwards Altman called as bioplast, then finally Benda has a name coined this mitochondria. This mitochondria, it is most important for the cell respiration. It is also we can call as powerhouses of the cell. వీటిని కణశక్తియా భాండాగారాలు, కణశక్తియాగారాలుగా మనం పిలుస్తాం. మైటోకాండ్రియా ఈజ్ ఆల్సో వీ కెన్ కాల్డ్ ఎస్ ఏ సెల్ ఇన్ ఏ సెల్. ఒక కణంలో గల కణాంగంగా కూడా మనం మైటోకాండ్రియాని మనం పిలుచుకోవడం అనేటువంటి జరుగుతుంది. My dear friends, remember this mitochondria is most important for the synthesis of the adenosine triphosphate ATP, and also it is present RNA, DNA and 70S type of ribosomes. Why? Because the structure of the mitochondria, when we are observed, the double membranous cell organelle, the outer layer, it is in smooth in nature, that in between inner layer and outer layer, the space we can call as perimitochondrial space. The inner layer, so many folds we are observed. These folds we can call as cristae. These folds is most important for the electron transportation system, for the synthesis of the energy. And the matrix contain abundant enzymes for the citric acid cycle, or the also we can call as the what we can say that Krebs cycle. Sir Hans Krebs are discovered this cycle. It is in aerobic respiration. Along with this, why we can call this cell a cell in a cell? Because the matrix contain the circular DNA structures, extra chromosomal DNA which is present inside the matrix of the mitochondria. This circular DNA, it is also we can call as mitochondrial DNA or empty DNA. This mitochondrial DNA or empty DNA, which is most important for maternal inheritance, మాతృత్వపు అనువంశికతలో తోడ్పడేటువంటి అత్యంత ముఖ్యమైనటువంటి DNA మైటోకాండ్రియల్ DNA. ఇందులో దీంతో పాటుగా 70S రైబోజోమ్స్ కూడా ఉంటాయి. మై డియర్ ఫ్రెండ్స్, రిమెంబర్ ఆల్ ప్రోకారియోటిక్ సెల్స్ దే ఆర్ possess 70S type of ribosomes, whereas all eukaryotic cells they are possess 80S type of ribosomes, even though the mitochondria also possess 70S type of ribosomes. Actually eukaryote possess 80S type of ribosomes. So that's why the mitochondria we can call as a cell in a cell, powerhouses of the cell, including along with this mitochondria, the mitochondrial DNA also synthesizes most important enzymes which are useful aerobic respiration. వాయు సహిత శ్వాసక్రియలో తోడ్పడేటువంటి అత్యంత ముఖ్యమైనటువంటి ఎంజైమ్లను ఉత్పత్తి చేయడంలో ముఖ్యంగా ఈ మైటోకాండ్రియల్ DNA అనేటువంటి తోడ్పడుతుంది. మాతృత్వపు అనువంశికతతో పాటుగా శ్వాసక్రియలో తోడ్పడేటువంటి ఎంజైమ్ల ఉత్పత్తికి తోడ్పడేటువంటి DNA మైటోకాండ్రియల్ DNA. అంతేకాకుండా మై డియర్ ఫ్రెండ్స్, రిమెంబర్ ది మైటోకాండ్రియా ఈజ్ ఆల్సో ఇన్వాల్వ్డ్ the PCD, programmed cell death. Actually we know that the lysosomes are involved in this programmed cell death, but even though the advanced concept, the mitochondrial dynamics, whenever we are going to observe, it is involved in the fusion, and fission also influence energy efficiency and also the stress tolerance from the external environment, and even though the internal biochemical reactions. The programmed cell death means the cell, whenever the cell is going to face any type of disease or attacked by any type of pathogen like bacteria, virus or fungal pathogen, automatically the cell by themselves go into death. So that we can call as programmed cell death, or also we can call as apoptosis. This is actually this process takes place by lysosomes, but in advanced concept by the research studies, the mitochondria is also involved in this the programmed cell death. This is a most important advanced concept. Then what about the plastids? We know that Julius Van Sachs first identified the 3 types of the plastids which are present on the all plants. Only plastids are present, plant cell only. These plastids 3 types. Number 1, chloroplast. Chloro means green. The green chloroplast which are involved in the photosynthetic process for the synthesis of the food material. In entire world, the one and only biochemical process to synthesize food material that is involved this cell organelle, that is chloroplast. Then second type of the plastids, that is the chromoplast. Chroma means color, plastids means they are involved in the different type of metabolic activities. The chromoplasts which are present on the external parts of the plants, like all colorful parts like the petals of the flower and the colorful ripened pericarp of the fruit and also different colors of the caused by the croton plants, and even though the tulip flowers, different colors caused by the chromoplast. Then third one, that is leucoplast. Leuco means colorless or white color. This leucoplast most important function, it is preserve the food material. Preserve the food material, maybe the carbohydrates, maybe the proteins, maybe the lipids. The carbohydrates preserved leucoplast we can call as amyloplast, whereas the proteins are preserved like alluroplast. Then finally, the fats and oil grains, oil contained grains are the preserved leucoplast we can call as elioplasts. So these three types of the plastids we are observed in the plants. The most important chloroplast structure we are going to discuss here. The beautiful structure, it is a discoid shape of the double membranous cell organelle, most important for the photosynthetic cell organelle, that is a chloroplast. This chloroplast contain two membranes, outer membrane and inner membrane. Between two membranes, that is we can call as periplastidial space. Inside we are classified into two different parts. Number one, the membrane structures that we can call as the thylakoid membranes. The thylakoid membranes, one by one, they are ఒకదానిపైన ఒకటి దొంతలుగా అమర్చబడి అటు ఒక ఫ్రెట్ మెంబ్రేన్గా ఏర్పడి మనకు గ్రానాగా లేదా పటలిక రాశిగా ఏర్పడుతుంది. దిస్ థైలకాయిడ్ టర్మ్ హ్యాస్ కాయిన్డ్ బై మంకే. ద సైంటిస్ట్ హ్యాస్ కాయిన్డ్ దిస్ thylakoid, that is scientist name Manke. Then after these thylakoids, the most important area, the dark reactions takes place, that is the stroma. The stroma, it is the most abundant source of the enzymes which are useful for the photosynthesis, especially dark reactions. The most important enzyme rubisco, that is also we can call as the abundant enzyme in the entire plant world. Rubisco, the abbreviation of the rubisco, ribulose 1,5 bisphosphate, carboxylase and oxygenase. It is also we can call as dimorphic enzyme. దీన్ని మనం ధిరూపక ఎంజైమ్ అని, రెండు రూపాల్లో ఉన్న ఎంజైమ్గా మనం రుబిస్కోను మనం పిలవడం అనేటువంటిది జరుగుతుంది. అలాంగ్ విత్ ఇట్, మై డియర్ ఫ్రెండ్స్, రిమెంబర్ ది stroma also contains 70s type of ribosomes along with circular DNA. My dear friends, remember mitochondria matrix possess the circular DNA within the eukaryotic cell of the plant cell and also animal cell. Even though the plant cell, the chloroplast also contains 70s type of ribosomes along with circular DNA. The circular DNA we can call as plastidial DNA or chloroplast DNA, ctDNA. This chloroplast DNA which is involved, it is involved in the synthesis of the rubisco enzyme. Half of the rubisco enzyme synthesized, and also the other enzymes related to dark reactions enzymes synthesized by this chloroplast. So this what is the advanced concept of this chloroplast? This is the plastid to nucleus signaling adjustment in photosynthetic genes, and also it is involved in synthesis of amino acids, fatty acids and secondary metabolites. Why we can call this the synthesis of the amino acids and fatty acids and secondary metabolites? Why? Because the 70s type of ribosomes along with the circular DNA or plastidial DNA which is possessed in the stroma of the chloroplast. So that's why the advanced concept told that the plastid to nucleus signaling adjust photosynthetic genes are the most important involvement of the molecular level.
Right, sir. Thank you. So double membrane to clear mitochondria and chloroplast. You said the cell within the cell.
Yes.
Mitochondria, cell within the cell, and chloroplast within the cell. And another important character is, apart from the nucleus, actually nucleus have the genetic material,
Yes.
DNA.
Yes.
Apart from that, DNA also have the genetic material. Chloroplast also have the genetic material.
Yes.
So the small discussion here, cell within the cell. Okay. Before that, the small connection, how the cell evolved.
Okay.
So evolution of cell actually first after the chemical reaction, that means the 3 billion years ago, the small chemical reaction takes place in water.
Yes.
First life begin in water.
Yes.
So after chemical reaction, first some genetic material, nucleic acid, RNA developed without cell.
Yes.
There is no cell structure.
Okay.
But later it is transformed into prokaryotic cell.
Okay.
Such prokaryotic cells are archibacterial cell, and aerobic bacteria. The what happened later, the archaebacteria cell engulfed this aerobic bacteria.
Yes.
Actually, these two different organisms.
Yes.
But what happened later here, the first archaebacteria cells engulfed aerobic bacteria.
Yes.
Later, when it is entered into the archaebacteria cell, these aerobic bacteria cells transformed into mitochondria.
Yes.
Right, sir?
100%.
So, that's why we called as a cell within the cell. And this transformed mitochondria having DNA and other 70S ribosome. Already we discussed, as sir said, prokaryotic cell have the genetic material DNA and 70S ribosome.
Yes.
The same genetic material present and 70S ribosome also present in the mitochondria. So, that is why called cell within the cells, right, sir? And another one is chloroplast.
Yes.
So, from the like that first that is the early eukaryotic cell developed. So, this is done by endosymbiosis process.
Yes.
Because the cell engulfed another cell, and what happened actually when entered into the cell, digestion takes place.
Yes.
But here what happened, instead of digestion, it is transformed and integrated within the part of the cell and transferred into mitochondria. In the same way, after formation early eukaryotic cell, this eukaryotic cell engulfed another photosynthetic bacteria.
Yes.
So, photosynthetic means the photosynthetic bacteria already have the chloroplast.
Yes.
But after enter into the cell, that means early eukaryotic cell without digestion. Actually, when entered into the cell, digestion takes place.
Yes.
But there is no digestion, but it maintains the mutualistic, that means symbiotic relationship within the cell and transferred as a chloroplast.
Yes.
That is why the plant cell evolved. So, this is secondary endosymbiosis.
Yes.
So, first endosymbiosis, early plant cell, early eukaryotic cell, later plant cell evolved. That is why here the cell within the cell, the called both mitochondria and chloroplast. Thank you, sir. Well explained.
Yes.
And it is very helpful to the all competitive exam, competitive preparation students also. Yes. Sir, apart from the double membrane, so please explain about these also very important, the single membrane cell organelles. What are those? And just give the brief about, just brief explanation, structure and functions.
Sir, actually, previously we have discussed about the most important cell organelles related to the photosynthetic and also the respiration.
Yes.
Cell respiration done by the mitochondria, the photosynthesis is done by the chloroplast of the all eukaryotes.
Right.
But whereas the bacteria, photosynthetic bacteria, they are possess the chromatophores, the special type of pigments which they are possess, like bacterial chlorophyll and chlorobium chlorophyll, which are involved in the photosynthesis to synthesize the food material in the process of photosynthesis. That is in the prokaryotes. But whereas in eukaryotic organism, some of the other most important cell organelles also we are observed in the endomembrane system, that is endoplasmic reticulum. This endoplasmic reticulum has first reported by or discovered by K.R. Porter in 1953. The endoplasmic reticulum, most important, my dear friends, remember the longest cell organelle in the entire eukaryotic cell, that is endoplasmic reticulum, which is from the nuclear membrane to the plasma membrane, which expand the cell within the cell from nuclear membrane to plasma membrane. So that's why the cell organelle, endoplasmic reticulum, we can call it as the longest and largest cell organelle from the nuclear membrane to plasma membrane. And these endoplasmic reticulum has classified into two types based on the functions of this endoplasmic reticulum. This endoplasmic reticulum, the main function is secretion. The secretion of the ribosomes, that endoplasmic reticulum, they are possess the ribosomes. Wherever the ribosomes are attached in the surface of the endoplasmic reticulum, that endoplasmic reticulum we can call as rough endoplasmic reticulum. Whereas the absence of the ribosomes, the membrane surface of the endoplasmic reticulum, that is in smooth endoplasmic reticulum. The rough endoplasmic reticulum, which is involved in the secretion of the proteins, and the smooth endoplasmic reticulum, the second type of the endoplasmic reticulum, which are involved the synthesis of the or secretion of the lipids or fats. This is the most important function of the smooth endoplasmic reticulum. Now we are observed the structure of the endoplasmic reticulum, the beautiful structure we are observed. The cisternae are observed in the endoplasmic reticulum, which is attached on the surface of the external nuclear envelope to the attached to the surface of the plasma membrane. So that's why the endoplasmic reticulum we can call as the longest cell organelle we treated. Then afterwards, the most important, the advanced concept of this endoplasmic reticulum after synthesis of the or secretion of the proteins and also the lipids, then afterwards the advanced concept which it is also involved detoxification. Whenever the toxic substances are synthesized or produced within the cell, or even though which is enter into the from the outside or as a foreign bodies, the smooth endoplasmic reticulum is acted as a detoxification. And also the endoplasmic reticulum which is involved stress responses. We all already discussed so much of external that is the abiotic stress like salt stress, cold stress, temperature stress. This is the external or the abiotic stress. Then even though the biotic stresses also we are observed inside the cell or inside the organism, due to that cause the endoplasmic reticulum they are involved and unfold proteins responses help in survival during environmental stress. Because of so many external changes we are observed due to the global warming and other the ecological factors, or even the ecological environmental pollutions, environmental changes, rapid changes we are observed due to the urbanization or other so deforestation, other regions. So that's why the endoplasmic reticulum is involved the protect from the external or environmental stresses. The Kumar sir already asked about the another type of the cell organelle, that is the Golgi apparatus. The Golgi apparatus also most important cell organelle. It is discovered by Camillo Golgi in 1898. The Camillo Golgi is also the most important function, that is the packaging material to be delivered either the intracellular targets or the secreted outside of the cell. So the Golgi apparatus involved not only for the synthesis of the proteins and also the other lipids, it is also important site for the formation of the glycoproteins and also the glycolipids. The glycoproteins or glycolipids are used or useful in the formation of the plasma membrane during the cell division of the plant cell. And in plant it involves the synthesis of the cell wall materials, and also they play a role, important role for the formation of the cell plate, for the phragmoplast formation during the cell division. Whenever the cytokinesis takes place, this phragmoplast produced by the involvement of the endoplasmic reticulum. Then the Golgi apparatus advanced concept, not only the packaging of the proteins and lipids, but also it is also involved the wall of the polysaccharides and biosynthesis and secretion of the defense compounds due to the cause of the so many pathogens, and also the production of the other the toxic substances within the cell. It is protecting as a defense compounds synthesized by the Golgi apparatus.
Continue with the lysosomes.
The another most important cell organelle, that is lysosome, which is present in all type of the eukaryotic cell, maybe the plant cell and also the animal cell. The lysosomes also we can call as the suicidal bags of the cell. These lysosomes are discovered by D. Dewey. D. Dewey was the scientist who discovered the lysosomes. These lysosomes are having the different types of the enzymes. The hydrolysis enzymes are present inside the lysosomes, which are involved the digestion, especially the metabolic processes, and also the secretion of the enzymes, and also the digesting compounds like carbohydrates, proteins and lipids, even though the nucleic acids also. And also whenever the starvation time, the starvation condition, the lysosomes release for the hydrolyzing process, the cell is going to death, and also also we can call as this we can call as programmed cell death, also we can call as apoptosis, also we can call as the autolysis. The autolysis has done by the lysosomes, which are present in all type of eukaryotes. Then vacuoles, which is also most important cell organelle, the most distinguished feature of the plant cell. All plant cells they are possess the younger cell it is possesses number of smaller vacuoles, but whereas the large vacuole which is present in the older or the aged cell, we can identify the vacuole is the larger size. The number is less, the size is increased in the aged cell or older cell. The vacuole, the most important vacuole, it is surrounded by tonoplast layer. The vacuole inside the vacuole we can call as filled with cell sap. The cell sap contains so many different types of the pigments like anthocyanins and also the other excretory products or excretory materials which is preserved inside the vacuole. And my dear friends, remember the vacuoles also important for the osmoregulatory process, even though some of the amoeba also the central vacuoles are involved in the osmoregulatory process. The most advanced concept of this vacuole, it is involved not only the store of the water and lipids pigments like anthocyanin pigments and secondary metabolites, and also the tonoplast transporters regulate ion balance and the pH, the hydrogen concentration, and vacuole also degrade macromolecules during autophagy. So autolysis or autophagy, that is nothing but programmed cell death. Then cytoskeleton, which is most important, it is cause of the special perfect structure due to the cause of the cytoskeleton. Due to the presence of the cytoskeleton, we are observed the microtubules and microfilaments and intermediate filaments. These are in the part of the cytoskeleton. It is caused the structure, definite structure and shape of the cell caused by the cytoskeleton, and also it is acted as a skeleton. It is also a framework. It is also acted as a framework. It is most important for the function of the during cell division. The cytoskeleton, which is formed the spindle fibers for the involved in the mitotic division or even though the meiotic cell division for the transport or the attraction or the contractile from the most important chromosome or chromatids. And the most important that is the cilia, flagella, these are the most important organelles which is involved. It is the external part which is involved for the motility or the movement from one place to another place for the protection, for the reproduction, for the collection of the food material. The most important structure of this structure of the flagella, 9+2 arrangement microfibrils we are observed in this and most important the final interorganelle communication. The chloroplast mitochondria, they are combinedly coordinate energy metabolism, whereas the endoplasmic reticulum and Golgi apparatus, both they are interact for protein trafficking during the most important process, that is the molecular level. The protein trafficking is controlled by this endoplasmic reticulum and Golgi apparatus. This is a interorganelle communication. Due to this cause, the biochemical and even though the secondary metabolites and all the molecular level functions are continuously without any interruption, they are going on due to the interorganelle communication. Then finally, we can going to conclude that the understanding advanced plant cell structure and organelle functions provide insight into how plants adapt to environmental challenges, optimize metabolism and maintain growth due to the sudden changes of the external conditions, environmental conditions, even though they can sustain and they survive from the external environment. Such knowledge is also essential for biotechnology. Nowadays we can observe, due to this biotechnology, we can produce the recombinant vaccines like COVID vaccine due to the presence of the involvement of the biotechnology, genetic engineering process, and also we can identify the knowledge related to crop improvement and stress resilient research. So this is the most important advanced concept of the cell structure and the functions of the cell organelles.
Thank you, sir. Thank you, Srinivas sir, and thank you, Thirupat sir. You are giving a wonderful lecture. Sir, you explained about the plant cell, cell wall, plasma membrane and nucleus, mitochondria all. The lack of time, we did not explain about those metabolic activities of cell organelles like mitochondria metabolic activities, glycolysis and Krebs cycle and ATP synthesis, ATP formation. Likewise, in chloroplast, how the photosynthesis takes place, light reaction and dark reactions. They did not explain the lack of time. So, anyway, thank you all.
Thank you, Kumar sir. Thank you very much. Thank you, Srinivas.